I'd like to introduce the Protein Maker by Emerald Biosystems. The Protein Maker is a versatile, high-throughput chromatography robot. The Protein Maker is designed for parallel protein purification of up to 228 samples per day. It operates in production, scouting, and complex purification modes with up to 24 chromatography columns running in parallel. It is made to process large or small sample loads and allow a user to set up a purification run and just walk away. The Protein Maker features 24 precision syringe pumps and an XYZ gantry that allows for pickup of reagents and delivery of purified protein to any container on the deck. The plumbing system includes a 9-port valve head on each syringe pump and a step mixer for step gradients. Samples can be loaded from either deck or tube rack. The Protein Maker's deck has 20 positions for 96 or 24 well SBS blocks and is shown here set up for 4-step protein purification. This is an example of the Protein Maker picking up a sample from the deck for loading onto the columns. This is the Protein Maker at work, collecting column fractions. The Protein Maker software is straightforward and easy to use. You select the protocol and number of run cycles, then set flow rates for each operation. It allows for automatic column stripping and equilibration between runs. The software also allows full manual control of each pump, valve, and gantry position. The Protein Maker is adaptable for many different applications, from parallel affinity purification, chromatographic scouting, antibody fragment purification, immunodepletion, and sophisticated two-dimensional purification schemes. One key application is integrated protein expression testing, purification, and crystallization. This shows purification of his-tag proteins with internal deletions, one of which was used to obtain the crystals shown. If you are doing a mutational analysis project, Protein Maker allows you to test mutants for expression levels and then purify them if expression levels are high enough. If a higher level of purity is needed for his-tag protein, the Protein Maker can carry out dilution of eluates, then reload and re-chromatograph the samples in a second round of metal chelate chromatography. Another common application is purification protocol scouting. In this application, several different columns are used to purify the same protein. These are some of the columns and buffers we commonly use with the Protein Maker. These are examples of some of the results you would see for 6 different columns during protein purification scouting. Another use for the Protein Maker is antibody purification. These are 24 Protein A antibody purification columns, as well as the software interface for this application. The Protein Maker can be configured to perform 4 consecutive unattended antibody purification runs comprising 96 individual samples. This slide shows the result of 4 consecutive purification runs. Another application is depletion of high abundance proteins from serum or cerebrospinal fluid samples with antibody columns. This shows the results of serum or depletion of high abundance proteins from cerebrospinal fluid. Here the Protein Maker is set up to run 2 Agilent immunodepletion columns. An additional application is a 2-dimensional purification protocol, wherein proteins are fractionated successfully on 2 different kinds of columns. In this application, protein fractions from the first column are delivered to and picked up from the deck for delivery to the second column. The technical specifications for the Protein Maker listed here are available for download at our website, www.emeraldbiosystems.com. Copies of this presentation are available for viewing or download as well. Thanks again for the team at Emerald Biosystems.